Well, it was my pleasure to introduce María Rebolleda-Gómez, and I hope she might connect during this presentation. So I had the pleasure to meet her in a few months ago, and even though we are working in different topics, we realized that there is like a lot of common ground in the end in the study of early life. Because in the end, most of the questions that we address require some kind of network understanding. Like we cannot really work with isolated systems as chemicals or species. We need to integrate those, understand how they are interacting with others. And that's what really will allow us to make our study of early life and the origin of life. Today I'm gonna I'm gonna make a little overview of like why networks are important and why. My perspective is a little bit more chemical than biological. I'm currently working on the description of prebiotic chemical reaction networks to explain, like trying to systematize the origin of life. When I think that in the end, well, maybe like life RCN is based not so much on the idea of trying to put research together to understand the origin of life, but more the development of life, of early life. But in general, like these two questions are very similar. So here I like to bring up there is this. In this slide, I like to bring up Aristotle. Like back in the day, like Greeks were already thinking like about the origin of life, and maybe their theories about the origin of life are not really that relevant today. But actually, their metaphysics might still be, because there's like this idea that they have about like how substance and matter how matter and form give place to substances. And in the case of biology, we have that, well, we have different matters, like in this case, bioelements, biomolecules, etc. But those are not really like, those don't make a living organism or don't make any evolvable thing. We need to integrate them in something else. In this case, for instance, we might talk about information polymers, metabolisms, etc. Okay, I'm gonna skip a few slides here. In the end, for instance, when we look at chemical systems, this is the prebiotic chemical systems. We have these collections of reactions that have been described to understand how we can create these biomolecules. That means how we can create the material that makes life, but we don't really have that much knowledge about what makes. I mean, like we don't know yet how to turn this into a living organism. So in the end, like the quest of trying to understand the origin of life might be, besides being related to understand how we build this form, like this material, might be also related with how we generate these behaviors from this material. Something similar might happen at the biological level. Like we might have different species, different proteins, etc. But if we don't integrate them into higher order systems as metabolisms, etc., or microbial communities, in this case, it's going to be difficult to understand what they are doing. So from my perspective, working on chemical systems, I find that a solution to integrate how these different molecules might give place to biotic behaviors is to integrate them in chemical reaction networks. That means like understanding how different chemical reactions require certain substrates and products, and integrating those into higher order chemical reaction networks, so we are able to generate systems that might look like metabolisms, etc., and that might have higher order emergent properties as the possibility of replicate, the possibility of evolving, etc. Here, this is a little bit different, as I say, from what Maria is studying, but I think that there is like as I say, there is like some common synergies between what we do, and I think that that's all related with what networks can tell us. We live on a connected world, and I think that I don't need to convince you of that. And like the idea of connections is something that might have been people has more or less known it like for a long time, but there was also a little bit of, okay, so what? Things are connected. And it's not until like 20 years ago that we started to discover what are the properties of connections and how the connections itself can generate new knowledge. So for instance, here, when I talk about networks, I'm going to talk actually about graphs. Like a graph in the end is the mathematical representation that we choose to talk about a network. So we know, for instance, in leaving aside biology, we know a very good examples of networks. For instance, we know the traffic systems for both road and air traffic transport. And although these are different, like those are networks that are more or less dealing with the same phenomenon. That means people moving to one place to another. Okay. Oh, Maria is here. Yeah. Okay. These have different properties. Like for instance, road traffic is way more. It depends on way more nodes to move to one place to another, while air traffic behaves more as something we call a complex network. And then we also have the social interactions, the internet, metabolism, etc. So anyway, I will really enjoy spending a lot of time talking about this. But I think we have our seminar with us right now, so I might go to introduce her. Okay. So as I said, I had the pleasure to meet Maria just a few months ago. Maria studied a degree in biology in the university in the UNAM, the National University of Mexico, and then she followed with a PhD in the University of Minnesota with Michael Trevisano on the evolution of multicellularity. Fascinated by the evolutionary dynamics, she did 2 postdocs. The first one at the University of Pittsburgh with Tia Lynn Ashman, focusing the plant microbial communities, and a second one with Alvaro Sanchez in the prediction of evolutionary dynamics. Now she runs her own group in the University of California, Irvine, focused on the prediction of community assembly, function, and evolution. As I say, I had the pleasure to meet Maria in person, and I really enjoyed my conversation with her. And I hope that you all enjoy today's seminar. But we'll dive into more like the biological aspects of network organization.
Hi, thank you so much, Bruno. And I'm sorry I was late. I totally calculated wrong the time, and I thought it was at 11. So sorry, everyone, and thank you for waiting. So I've been really excited to present in this seminar in particular, because I think there's a lot of the work that in astrobiology and in general in geobiology and paleontology has really inspired a lot of the questions throughout my career. And so I was excited to basically share my work. And so without further ado, let me present. Okay, so I'm interested in innovations and constraints, and I see these as sort of 2 faces of the same implications of adaptive landscapes, and I'll get more to that. And specifically, lately, I've been really interested in what we've been calling metabolic rules. So these are very simple kind of metabolic constraints or little changes that really that generate holistic rules that then we can use to predict community dynamics. So these will all become clearer. And in general, I'm really interested in history, and so how past changes affect present and future. How does past evolution affect present and future ecological and evolutionary dynamics? And if past can past changes help us predict future dynamics? And so I don't think I need to convince anyone in this room that the evolution of certain innovations like photosynthesis have really changed our environment, have changed our planet, and continue to shape current evolutionary and ecological dynamics in important ways. Furthermore, some changes depend a lot on the past. And so these are two very different animals, a bird and a marsupial, that have occupied pretty much the same niche. They eat little larvae and worms in the bark of trees. But because of their evolutionary history, how they do it now, it's very different. So the woodpecker uses its beak, but the nine-nine doesn't have a beak, and so it uses a finger, and this long finger has evolved in a sort of similar way than the beak, but it's constrained by the whole sort of morphology of the organism and the rules of that morphology. And so in some ways, we tend to think that history does help us predict the present. And so in the words of William Shakespeare, what is past is prologue. But we also know in evolution that historical contingencies can hinder predictability. So we might have that if there was no historical contingency, no matter where you were in the past, future selection would take you to the same point in this first scenario. But given that we have prevalent historical contingency potentially, then different starting points take you to different outcomes, and that makes it much harder to predict. On the other hand, historical contingency also restricts the space of possibilities, because if I started in this light blue state in the top, then I can only evolve these red possibilities, and maybe these yellow possibilities are no longer available. Whereas if I'm this dark blue, I'll evolve these yellow possibilities, and these red pinkish possibilities might not be available anymore. And so in some ways, past evolution might take me in particular trajectories and close others. And so that is a little bit what I mean by saying that innovations and constraints are in some ways two sides of the same coin, because we get these big evolutionary innovations, and those take us into particular paths, and they will have their own constraints. And once we're constrained, these constraints limit the space of what's possible. So imagine in this schematic, we have an axis of performance on trait X, and then an axis of performance of trait Y. And if I didn't have any constraints, then all these space is available. But because there's some constraints, this red space is actually not available, and that limits all the possibilities and makes evolutionary biology a little bit more predictive. So it also, these constraints affect the topology of adaptive landscapes. And so the interactions between traits are precisely what creates these peaks and valleys, the interaction between fitness of different traits. And so these we have these constraints, and so we might be able to occupy this sort of Pareto front, and selection will push us up to there, but that's where we're stuck, right? And at that point, we cannot move farther. And so often evolutionary innovations are the kind of changes that break or change these constraints, and they allow us to explore this space. And with that, it completely changes the dynamics. And now we don't actually have that constraint if we were able to break it. But of course, accompanied with any major change like this one, there will be new constraints and trade-offs to contend with. So let me illustrate this with an example, because so far I've been talking in these very abstract terms. And the work I'm going to present in this example is work I did in my PhD following experiments that William Radcliffe and Mike Travisano started in the lab, and then work that Mike and I did. So there are constraints limiting the maximum size of a cell, right? So diffusion, for example, diffusion to increase volume to surface area. And so a cell cannot get infinitely large without very complex mechanisms of active diffusion and other things. But else being equal, a cell has big constraints on how big it can get. But if we select for size, we take these yeast cells and select them to get bigger. So we put them in an environment where large size is highly favored, in this case, by letting them centrifuge in a tube, letting them settle in a tube, and just taking the cells that settle first. Then what we have is that in this case, 10 out of 10 populations evolve some form of multicellularity. And so multicellularity is this innovation that breaks this constraint, this trade-off of size, because now we can actually get much bigger than a single cell could. And that's why in this setup, in this experiment, we get readily the evolution of multicellularity. But then once we get the evolution of multicellularity, multicellularity will pose new challenges. And so increased size comes with increased costs of local competition in the absence of dispersal mechanisms. And what I mean by this is when we compete the multicellular strains with their unicellular ancestor, both in liquid or on plates, without the selective pressure for size, so we are not selecting for settling anymore, what we get is that in well-mixed environments, multicellularity is costly, and that's because of growth, and I'll get more into that. But it's especially costly in spatially structured environments. So let me get a little into that. So we have these two environments, well-mixed environments, where all the resources and cells are everywhere, and then these structured environments where organisms only have access to local resources. And so there is a fitness cost of multicellularity when grown in well-mixed environments. So this cost of being in liquid. And that cost has to do with differences in growth. So here I have in gray, multicellular growth rates at different concentrations of the media. And then in black, the growth of the unicellular ancestor. And as you can see, at all concentrations, the unicellular ancestor grows better. And this is because in these multicellular clusters at the center, the cells are not getting resources and are not reproducing. And so growth rate and cell division is going to be much faster for the unicellular. However, when we do this competition, we see even more, we see that multicellularity is even more costly in these spatially structured environments where actually resources are all localized. But when we do growth, we don't see any difference between single cells and multicellular organisms in these plates. And that kind of makes sense because a single cell is going to grow like a colony in these plates, and it's going to look very similar to a multicellular aggregate where the cells inside at the center or inside the colony are not getting resources. However, when we compete them, we see a cost of fitness. So why is that? Well, that actually has to do with dispersal and the fact that in this case, our multicellular organisms have not evolved dispersal yet, so they're constrained by very intense local competition. And what I mean by that is, let's imagine we spread the same number of cells and cells in multicellular individuals in a plate, and we let them grow. And so our single cells are growing slower because at each point where they landed, there's less cells, whereas the multicellular ones very quickly reach sort of their carrying capacity. But by then, they cannot move out because the single cells are occupying all the other resources. And the single cells can keep using those resources and immediately grow. So the multicellular organisms immediately have a lot of cells around them because they are a lot of cells together that limit how much resources they can acquire. And so these evolutionary innovations, the evolution from unicellular to multicellular, changes the set of constraints and opens new opportunities. So multicellularity opens tons of new opportunities, but it also generates its own constraints and its own selective pressures and it changes this adaptive landscape. And so what I find is very interesting is that evolutionary innovations, these new big changes actually lead to high historical contingency. So not all single cells are going to evolve multicellularity, but once you evolve multicellularity, the effect of mutations afterwards are going to be very different in a multicellular background than they were in the unicellular ancestor. And so past history, past changes are going to have an intense effect on future dynamics. And so any change, I feel like evolutionary innovations tend to be these changes that really shift the adaptive landscape. And any change that's really shifting the adaptive landscape is going to lead to high historical contingency. And so something I've been getting really interested in is trying to predict evolution and ecological dynamics. And there's many reasons we might want to do that. So as our planet is rapidly changing with climate change, we want to predict how microbial communities, for example, will adapt and respond to this change. And of course, we would like to predict how things like COVID, how are they going to evolve? What's going to be the next strain? What traits will it have? But microbes live in very complex communities. So these are some images from some beautiful work of Ramirez Puebla et al. I had nothing to do with this, where they took microbial biofilms from kelp. And what we can see is that there's tons of different organisms living very close together, and they have very defined spatial organization. And so in these communities where there's a lot going on, it might be very hard to predict any dynamics, and especially because historical contingency is affecting both the ecology and the evolution of all of these cells. But as I said before, maybe we can use constraints that might be physical or physiological to limit the space of possibilities and have better predictions. And in particular, metabolism mediates bacterial interactions, so it mediates the ecology. And we might think that new metabolic structures might be hard to evolve because of pleiotropy. So what I mean by this is, of course, things very at the edges of the metabolic network might be kind of very simple to evolve. You might just get a gene from horizontal gene transfer or a little mutation or something, and that can be readily evolvable. But when you have something in the central metabolism or something that's highly connected within the network, then any change that happens there is going to have downstream effects all over the rest of the metabolism, and that might have high pleiotropic effects. And so because of that, it might be fairly constrained. And so these constraints could increase the predictability of both ecology and evolution. And that's what we mean in saying that metabolic rules then can help us understand and predict changes in microbial composition. So to evaluate this, when I joined the lab of Alvaro Sanchez for my postdoc, what they were doing in the lab were these experiments of replicated community assembly. So basically, for those experiments, you just take microbes from different sources. And what you want is sources that have a high diversity of microbes, so that then you can do a community assembly selecting from a sort of big pot of variation. And then you grow them in a simplified defined environment, and you transfer them over a few generations until the communities stabilize. And so what you get is that some microbes grow better, they have their own interactions, they're producing different metabolites, and you get a new state of the community. And so then we can do this many, many times and in different environments and ask how the environment is shaping the communities, how reproducible this assembly is, how predictable in some way. And so when we do this multiple times, we see that at lower taxonomic levels, so ESB or 16S variants or species, we see a lot of variation. So these are actually all replicates. So each of these bars is the relative abundance of different bacterial ESBs. And each of these bars is a different community, but they are all assembled from the same source. So coming from the same exact pool, we get tons of variation in different communities. But at the family level, they look pretty reproducible, and we can actually even predict quantitatively this ratio of this light blue to beige, which I'll talk more about. And so we can do this with many more communities, and what we get coming from this time from different sources, and what we get is this fairly predictable ratio of Pseudomonadeaceae or Moraxulaceae to Enterobacteriaceae and Aeromonadeaceae. And so why is this ratio predictable at this higher level? What's shaping this? And it actually has to do with the architecture of central metabolism. So the way the central metabolism is wired, it actually really constrains the efficient. It has two directions, a glycolytic and a gluconeogenic direction. And it's really, you cannot really do both at the same time efficiently, because if you're doing both at the same time, what you are actually doing is increasing the concentration of, for example, here, if you have a lot of glucose 6-phosphate, what you're going to have is that then the reaction is moving in this way. But if you're also producing the next thing here, then you actually don't have a gradient of concentration that fuels the reactions. And what you start getting is a lot of futile cycles. And so it's not really efficient to do both at the same time, which actually generates a strong pressure to specialize in either one direction or the other if you are using mostly the central metabolism for your carbon acquisition. And so in this case, Enterobacteriaceae and Aeromonadeaceae are families that can do fermentation in aerobic conditions, and they're glycolytic specialists. Whereas Pseudomonadeaceae and Moraxulaceae cannot, except for very few exceptions, do fermentation in aerobic conditions, and they have these gluconeogenic specialists. So they specialize in amino acids and organic acids, which tend to get metabolized in the gluconeogenic direction. And so this is some work that Jean Bille led that I was part of. I was lucky to be part of, where basically we grew a bunch of different gamma protobacteria, so Enterobacteriaceae, Aromonaceae, Pseudomonaceae, and Maracinaceae, the families that we kept seeing in our communities. We grew them in different carbon sources. And what we see is this very clear specialization in different carbon sources. So up here, the Enterobacteriaceae, these are all sugars, and they grow much better in sugars than Maracinaceae or Pseudomonaceae. And the opposite happens when we are starting to look at gluconogenic carbon sources. And so then we get much better growth in organic acids for Pseudomonaceae. And we're not the only ones that have seen these. This seems to be a fairly common pattern. And so Matti Gralka, Scholl Pollack, and Otto Cordero took a bunch of bacteria from heterotrophic bacteria from the ocean, and they grew them in many different metabolites. And then when they plotted in this principal component plot, what they get is that the principal component 1 is mostly an axis separating these gluconogenic from these glycolytic from these gluconogenic sources. So we get these dimensions where organic acids and sugars generate a very strong axis of specialization. And the evolution, we see that that this is mostly conserved. There's very low conservation at the species level, but then at between genus and order, so around the family level, is where it really starts being more conserved. These strategies, that's where they evolved this specialization, and that's why in our communities we see predictability at the family level, because we are basically segregating these communities along these axes. So what's happening is that our fermenters or glycolytic specialists, when they grow, they produce organic acid as a result of the fermentation. And the faster they grow, the more fermentation they do, and the more organic acids they secrete. And so over time, what we've seen, this is work that Sylvie Strela led, that I was part of. And what we saw there is that as time goes on, our fermenters are growing, they're consuming the glucose. This is in media that only contains sugars as the main carbon source. And then they secrete organic acids, and the respirators consume these organic acids. So that's what's shaping our communities. Have glucose. Fermenters can use that glucose to either make biomass or secrete organic acids. And then the respirators consume those organic acids to make respirator biomass or produce CO2. And we can, from there, get this ratio of respirators to fermenters. And we actually, what's really cool about this is we can measure all these parameters without actually having species interact. So we can give fermenters glucose, measure how much biomass they produce per molecule of glucose, measure how much organic acids they produce for molecule of glucose. And then separate from that, we can give the respirators organic acids and see how much biomass they produce with that. And from putting those numbers together, we calculate a ratio of respirators to fermenters of 0.31 versus our observed ratio of 0.29. And so these obviously are statistically not different. And so we can quantitatively, from just knowing this very simple metabolic structure, we can quantitatively predict the ratio of one metabolic group to the other. And so this was really exciting. It was high predictability. It seems like this is a trait that's shaping a lot of dynamics of heterotrophic bacteria in aerobic conditions. And so we were excited to look at this deeply and see if it really is predictable, then I should be able to disturb it and still be able to predict what's going to happen. So I should be able to push it and perturb and still get still be able to predict what's going to happen. And that's what we were thinking. And so this is work I led, and these are all the people that were working on it. And our idea was, can we use these simple metabolic constraints to predict community assembly along a temperature gradient? And so the reason we chose temperature is because temperature affects growth in a characteristic manner. And so as temperature increases, growth rate increases up to an optimum, and then growth rate decreases faster. And so we had a relationship between temperature and growth rate. And we also have a relationship between growth rate and acetate concentration. So we thought, well, then we can probably map temperature to acetate secretion and be able to predict that way the respirator to fermenter ratio with the model that we had. And so our idea was that with increasing temperature, fermenters are growing faster but more inefficient. So they're producing less biomass per molecule of glucose because more of the carbon is going to organic acids. And that allows for respirators to consume those organic acids. And therefore we have an increased respirator to fermenter ratio. That was the prediction. So basically, very quickly to go slowly at that is, when we have low growth rate, we should have low organic acid production and a low respirator to fermenter ratio. And as growth increases, you should have more organic acid production and a higher respirator to fermenter ratio. And at very high temperatures, we didn't know. On the one hand, we have less growth, so maybe less organic acid production. On the other hand, they're dying, they're stressed, they might be particularly inefficient and produce high organic acids. And so what we saw is, in different sugars that we tried, we get that as temperature increases, our fermenters are producing more organic acids until very stressful temperatures where they go all over the place. And actually, when we divide by the number of cells, we get that the per capita organic acid secretion increases more or less linearly with temperature. And consistent with this, we also see that the proportion of respirators to fermenters increases with increased temperature. And so these are all the carbon sources where we did community assembly. And in most of them, we see the same increase of respirator to fermenter ratio with increasing temperature. And that can be seen here. So if we ignore 12, which I'll get to, from 22 to 42 degrees Celsius, we see an increase in respirators to fermenters. And there's a few exceptions here and there, which are why not all lines are going up. So what's happening at 12, where we actually expected it to be basically only fermenters, and we see a coexistence of the two types and actually a lot of diversity. Well, our hypothesis is that at these cold temperatures where we have low growth rate of everyone, then all taxa are functionally kind of respirators. They're not producing a lot of acetate, and they have very little fitness differences. And so we have something close to neutral coexistence because everyone is growing kind of poorly. And we see that, right? We see that if in this plot, I have the difference in growth rate at each temperature in maximum growth rate at each temperature between fermenters and respirators. And we get that at 12, that difference is very low. It exists. The fermenters are still growing faster in sugars than the respirators, but it's a very small difference. And they're producing almost no organic acids into the media. So from this work, I think what we can see is that the evolutionary history of metabolism, these constraints generated by the architecture of the central metabolism, and then the specialization in glucogenic and glycolytic groups can help us predict ecological dynamics of community assembly. And so I think because I want to be mindful of everyone's time, and I was late for this, sorry for that, I'm gonna leave it here, but the short message here is we wonder how robust these metabolic rules would be to short-term evolutionary change. And this is work I did with these amazing collaborators. And what we saw after evolutionary is that overall, the ratio of respirators to fermenters is maintained over evolutionary time. There are clear perturbations. The second one was a snow day that we couldn't go to the lab. And so actually the transfer took 1 day extra. And what's interesting is everyone responded in, all the communities responded in the same way to the same perturbation, which is cool in its own way. But the first two perturbations, the one where we get a significant increase in RF ratio and one where we get a significant decrease, those two do have to do with changes, with evolutionary changes, but over time, those changes compensate and the RF ratio goes back to its original level. And these RF ratio of the evolved communities, the secretion of organic acids from the evolved communities actually predicts really well their RF ratio. And so, as I said, evolutionary history of metabolism can help us predict ecological dynamics of community assembly. And these dynamics are fairly robust, even in the presence of short-term evolutionary change. And I think the overall take-home of this talk is that evolution of metabolic architecture really constrains ecological and evolutionary dynamics, providing simple rules sorry, to predict current and future changes. And that when we have these big innovations, for example, in multicellularity, or in this case, innovations in metabolism, these should generate hard to predict dynamics. They should really change the adaptive landscape, and they increase historical contingency. Without further ado, thank you, everyone. As Bruno said, I'm starting my lab. I've been here a year, almost 2 years, in the Department of Ecology and Evolutionary Biology at UC Irvine, and I'm looking for a postdoc to study the role of spatial structure on metabolic division of labor. And we're going to be using directed evolution of microbial communities, which is another line of work I've been really interested in. And so please contact me if you're interested. And with that, there's a lot of people I want to thank, my colleagues, everyone in Alvaro Sanchez's lab, everyone in my current lab, all the administrators, janitors, drivers, everyone that makes our work possible. Thank you.
Great. Thank you, Maria, for your insightful seminar. Well, I think that the floor is open to questions. So,
and I see some questions in the chat.
Yeah, there's a first question. Okay, I'm going to go a little bit in a chronological order. So we have a question from Clausel. It's like, can evolutionary constraints be also overcome through convergent evolution to meet adaptation to similar environmental pressures?
Yeah, so that's what's, I mean, that is one thing that's interesting. So I think the farther the starting point, the more different the constraints are, the weirder that convergence will be. So I have that case of the marsupial using a finger and the woodpecker using its beak. So those two, the selective pressures are the same. They're overcoming some constraints, but what exactly are the constraints is very different depending on the starting point, and so the outcome looks very different. If the two things were sort of architecturally more similar in their morphology or whatnot, then the constraints look very similar, the selective pressures are the same, and then you get to stronger convergence. So that depends very different that depends a lot on where we started in the starting point.
Okay, maybe I can move to the next question from Agayan. Like, do you also check the genes that are responsible for intracellular proteins or other signals for cells connection?
Yeah, so in the multicellular case, there's some work, some unpublished and some published, where what we know from these multicellular organisms is the way they evolve the multicellular yeast is each population has a slightly different mutations, but all of them are mutations that basically prevent. So the way yeast divides is they form a bud, and the bud grows, the budding cell grows, and then it buds off the mother cell. And this separation of mother and daughter cell, all the mutations involved were involved in that separation. And so basically it's not letting the mother and daughter cell separate, and they keep growing that way. There doesn't seem to be tons of communication between cells at this point, although I would really recommend checking the latest work of Will Radcliffe, where this multicellular yeast has gotten like humongous, partly related to oxygen constraints. We were not able to do that, but he figured it out. And in that case, there's starting to be more communication between cells and other things.
Cool. Okay, we have another question from Russell, but we'll go back at it by the end. From Donald from Burke, have you tried perturbing the system by introducing altogether new metabolic lifestyles, such as methanogenesis, methanotrophy, photosynthesis, or something else that is orthogonal to the air-off dichotomy?
Yeah, so we haven't quite, we've been interested in, right now we've been looking mostly at carbon metabolism, and we've been interested in the interactions between phosphorus, nitrogen, and carbon metabolism in these dynamics. And so what happens if we have different ratios of C2N to P? So that's something we've been doing the experiments. Shin Sun, who helped me with the temperature, is leading that work. I'm not updated in the latest results of that system, but I think it would be interesting to, like, those are not completely orthogonal. In some ways they interact very clearly, but it would be very interesting to try something else. And I've been interested, like, we've been studying mostly carbon heterotrophy in aerobic environments, but obviously that's a tiny sliver of what metabolism exists on the face of Earth, and how bacteria interact. And so, yeah, we've been interested in including other axes. Thank you. So Hunter Dooley says, in your analysis of microbial communities, you show that carbon is
Sorry,
higher. Oh, sorry.
Sorry, that's why. Yeah. Sorry, you can go keep on. Yeah.
No worries. Family level based around its cycling. Do you believe that constraints around other elements, nitrogen, sulfur, phosphorus, etc., may have similar rules that dictate the presence of organisms at lower levels, genus species? I think that it will be different. So carbon metabolism is, especially at this level, is extremely conserved. Not all metabolic pathways have the same level of conservation. And so, for example, the pathways of denitrification are much less conserved in some ways. And so I think the functional predictability of the communities will be the same, but the conservation level will be very different, depending on at what level that metabolism is conserved and when did it evolve and how evolvable it is. And so I would expect we'll get much more predictability even at lower levels with things that are very, very conserved even at low levels, and less so with things that might be more evolvable. Yeah.
Good. I'm gonna come back to the question of Klaus. Oh, we have another question. Okay. Yeah, I'm gonna come back to Klausel. He asked, in the glycerol fed system, did the temperature results have a lower RF variation?
Yes, they do a little, and so I can maybe share again. Quickly. So actually, it's hard to see the colors, but this pink one is glycerol, and all the sugar alcohol, so this one is galacticol and sorbitol, those are the ones that don't have as clear of a relationship between temperature and the RF ratio. And we tend to see a slightly lower respirator to fermenter ratio in general in those in sugar alcohols. Yeah.
Good. And I have a question of my own. So I see like this feels quite similar to me, like how the eukaryote cell works somehow, right? Like the mitochondria is doing the respiration. So I was wondering, can you find that RF respiration fermentation ratio somehow also in eukaryotic cells, like the fraction of respiration fermentation that they are carrying out?
That would be I haven't looked, but that would be very interesting. Precisely because I suspect there can be a little more division of labor there, which division of labor tends to break these trade-offs, these constraints. But it would be interesting, especially in cells where all energetic metabolism has been totally shifted to the mitochondria, then you again have that trade-off, which in many multicellular eukaryotes, that's the case, but then they have different cells, and some might be doing some, like in our muscles, some will be respiring while some will be fermenting in some ways. So yeah, I haven't looked, but it would be interesting. Yeah.
Great. Okay. Well, I think that we don't have more questions. So, well, thank you, Maria, for this great seminar. and thank all of you for attending this our seminar series. Like, we will come back in July 10, I think, with Paula Wallender, if I'm right. So stay tuned, and thank you all for joining.
Yeah, thank you. And sorry again for the time confusion.
That's okay.
I bet it was very stressful for you, Bruno. I'm sorry.
No, I'm a good improviser. So.